Hello everyone, my name is Zuzana Pinkosheva, and I'm going to present our paper Cortical Activity of Graded Relevance. This work has been completed as a part of my PhD within the Laboratory X group at the University of Strathclyde with my co-authors and PhD supervisors Dr. William McGowan and Dr. Yashar Mushfeghi. This is my first SIGIR conference, and I'm very excited to be here to have this wonderful opportunity to meet the SIGIR community online and of course to tell you about our research. I'm going to start with the introduction, where I will define the key terms from the title, starting with relevance. Relevance is a central notion in information retrieval. It plays a key role in the user-system interaction, and it is an important indicator of system effectiveness and performance. But what exactly is relevance? Well, we don't know yet. Some refer to relevance as you-know notion, meaning that everyone knows what it is, and it doesn't have to be defined. However, the universal definition of relevance doesn't yet exist. This could be attributed to the fact that studying this phenomenon can be challenging. Relevance is considered to be multidimensional, dynamic, complex, difficult to quantify, and depending on users' perception of information relating to specific information need. Hence, to gain a better insight into the nature of this internal and subjective process, it is crucial to examine the underlying behavioral, physiological, and psychological mechanisms involved. Relevance should be considered as intersubjective, systematic, and measurable. This premise has been recently supported by empirical evidence of previous studies investigating relevance through the measurement of neurological signals. The effort of these studies has resulted in a significant contribution towards our understanding of relevance. This slide represents an overview of approaches to study relevance from a neuroscientific point of view. Results of these studies employing brain imaging suggest that overall, neurological signals differ significantly during the processing of relevant content versus non-relevant content across individuals. These studies can be split into two groups. One group which consider relevance as a part of the information retrieval, so consider the information need, and the other group that did not. I will start with the work of Mushfeghi and colleagues, who localize the brain activity differences during the processing of relevant versus non-relevant images. This study was the first one to examine relevance from a neuroscientific point of view within the context of information retrieval. The authors found that processing of relevant versus non-relevant images resulted in the brain activity differences, which were the most apparent in temporal, frontal, and parietal regions. A later study of Mushfeghi and colleagues employed also fMRI to understand the relevance in the context of textual information. The authors identified the following distinct regions: inferior parietal lobe, inferior temporal gyrus, and superior frontal gyrus that are crucial during relevance assessment. The increased activations of these regions for relevant items might be related to the complex cognitive processes such as memory retrieval, attention, and decision-making. Allegretti and colleagues employed the electroencephalogram, or EEG, in order to understand when does relevance happen in the brain. Hence, they were interested to investigate the relevance assessment process in terms of its timing. The authors found that when judging relevant images in the context of information retrieval, the peak difference in the processing of relevant versus non-relevant content occurs within the 500 to 800 milliseconds of stimulus onset. This time interval is highlighted in the figure, depicting the topographical plots of significance and difference between relevant and non-relevant condition. Kim and Kim explored relevance using video skims and found that the ERP components N400 and P600 have been shown to be important indicators of relevant and non-relevant judgments. I will talk about ERP components later in this presentation. A recent study of Yakuchi and colleagues found that relevance can be predicted in real time from EEG brain signals and eye movements during the IR task while the user engages with the system. Another direction of research examined relevance in the context of word associations employing EEG. For example, Oyster and colleagues found that peak significant difference between processing of relevant and non-relevant words was detected in the PZ channel after 450 milliseconds, maximizing at 747 milliseconds. Later, Oyster and colleagues introduced brain relevance paradigm, which enables recommendations of information without any explicit user interaction based on EEG signals evoked by the user's interest towards textual digital content. The mentioned study treated relevance as a binary variable. While such binary division is prevalent, seminal theories have proposed relevance as a graded variable. This is because treating documents of different relevant grades as equally important might not often reflect the user's interaction with the information. Recent research supports the idea of categorical thinking, suggesting that users divide retrieved results into 3 to 5 categories based on relevance. Additionally, it is important to focus on graded relevance, as some studies suggest that graded relevance, when compared to binary, improves ranking functions. Despite the fact, we still don't know whether there are clear neurological manifestations of graded relevance in the human brain. Therefore, there is no neuroscientific evidence suggesting that relevance could be in fact graded. Hence, for this study, we decided to investigate this research question. We wanted to understand whether the information relevant to different degree, that is in our case, non-relevant, low-relevant, and high-relevant, is also perceived in the brain in a different way, and if so, what could it tell us about the cognitive processes underpinning graded relevant judgment? I will now talk about the methodology to explain what approach we took on to investigate our research questions. We captured brain activity using 128-channel geodesic sensor net, which can be seen here on the picture. What is EEG though? EEG is used to record brain activity, and this is possible because our brain cells communicate through electrical impulses, so the EEG records the electrical activity. During the EEG assessment, sensors are attached to the scalp and face to pick up these electrical signals. EEG can therefore be used to determine changes in the brain activity. In terms of the design, we utilize a wet and subject design in which the participants perform question-answering task. During the task, participants were presented with a question and either relevant or non-relevant answer. The answer was presented word by word, and participants were instructed to make graded relevant judgment at each point of the answer as it was unfolding to them. We controlled the number of relevant versus non-relevant questions and answers presented to the participants, but we did not control the number of words that each participant has seen. The graded relevant judgment had 3 levels: non-relevant, low-relevant, and high-relevant, which was our independent variable, and our dependent variable was the EEG signal. Moving on to participants, we recruited 29 neurologically healthy participants in total. 4 participants were recruited for a pilot study to test whether the study runs smoothly, and their data were not included in the final analysis. 25 remaining participants were between 19 to 39 years old. 15 were females and 10 were males. From these 25 participants, we excluded 2 because of the high number of artifacts present in the EEG signal. In terms of stimuli, we adopted a question-answering dataset developed and used by Moshfeghi and colleagues in 2016. We have chosen this dataset since it had been widely used in the previous studies investigating IR phenomena from neuroscientific point of view. This adopted dataset was further expanded through the selection of additional questions and answers from Trek 8 and Trek 2001. Question-answering datasets are available upon request. We selected these questions to cover a variety of topics, such as politics or geography or sport. So let's talk about task. First, participants see the question, then the fixation cross indicating where the words of the answer would appear. Many of you know that in a task like this, while using EEG, it is important to limit eye movements to avoid eye-related artifacts in the data. Hence, we presented the answer word by word. For example, participants in the question, what is caffeine? Then the answer would appear word by word on the screen, that is, caffeine, A, stimulant, and so on. Once participant has seen enough words to determine whether the answer was relevant or non-relevant to the question, they had an option to skip the rest of the sentence and move on to the next step, where they provide a graded relevant judgment retrospectively to the words presented in the answer. So here participants were asked how relevant was, for example, a first piece of information, caffeine, and the answer, and then caffeine A, and next caffeine A stimulant, and so on, as the answer was unfolding to them. This is very important as we were not interested in the relevance of individual words to the answer. So in our analysis, we linked the brain activity recorded during the initial word presentation with a graded relevant judgment. We were also interested in the P300, N400, and P600 components, mainly because these components have been previously explored and associated with relevance, however, as I mentioned, with the binary nature of relevance. I will now talk about these components in more details to explain their importance in the relevance processing. So usually the letters P or N indicate whether the component has positive or negative deflection, and the number indicates whether the component is usually elicited for stimulus in terms of time. For example, the P300 is P hence positive and 300 as it occurs 300 milliseconds post stimulus. P300 is important as its amplitude has been shown to be proportional to task relevance and to the new and useful information. The N400, based on findings of Kim and Kim, is specific to the mismatching process for topic irrelevant information. Kim and Kim reported in the same study that P600 is specific to the process of updating context for topic relevant stimulus. We calculated the mean amplitude for each component for a specific time window, and we have selected these time windows based on previous literature. When visualizing these components for each condition, we observe that there are differences in the waveforms, and we use repeated measures ANOVAs to test whether these differences are indeed significant. The table on the right shows how many words participants perceived as non-relevant, low-relevant, and high-relevant. So what did we find? The take-home message and the main finding of this study is that there are significant differences with judging information as having high relevance, low relevance, or no relevance. Differences were present in all components of interest, so that is P300, N400, and P600, which might suggest a presence of variety of distinct cognitive processes that are underpinning graded relevance evaluations. This and next slide will show the waveforms for each component and condition. So blue is for non-relevant, red is for low relevance, and yellow is for high relevance. In grey, we highlighted the time interval of interest. So what do significant differences in amplitude of these waveforms tell us about graded relevance? For the high relevance, P300 amplitude was the largest, which might reflect the greatest attentional engagement. Hence, participants, on average, pay the most attention to the highly relevant information. Based on the findings of previous studies as well, the amplitude is proportional to the quality of information transmitted, the quality of useful information, perceived relevance of the information to the participant, and perceived relevance to the task. For the N400 component, the amplitude is the smallest for the high relevant condition. However, in this case, it is important to note that as the N400 is a negative deflection, the higher the amplitude means also the more negative amplitude. Small amplitude for the high relevant condition can be likely explained by semantical alignment of the information to the question. The greater mismatch between the information offered and the information provided in the question, the greater the amplitude, which might explain why the information judged as non-relevant elicited the highest amplitude. The most interesting condition was the low relevance in this case. This is because in this condition, N400 component more closely resembled non-relevance, suggesting that low relevance may be initially perceived more like non-relevant information until a certain critical threshold is reached. However, in terms of P600, low relevance was more similar to the high relevance. This might suggest a dual nature of the stimulus evaluation. So to conclude, what did we find? Our study is a support for graded relevance. We observed clear differences in neural activity when information segments were perceived as high relevant, low relevant, and non-relevant. The P300, N400, and P600 ERP components differ due to perceived relevance of the answer, but suggest the engagement of distinct cognitive processes during high relevance, low relevance, and non-relevance. Thank you very much for your attention. I hope you enjoyed this talk. I'm more than happy to answer any questions that you may have, and I'll be glad to get in touch. Thank you.